Speakers today are, we've got Stephanie Sopow from, it's really bad. She's the leader for Scion's Plant Health Diagnostic Services and curator of the National Forest Insect Collection. So she'll be talking today about the willow honeydew and a challenge for New Zealand beekeeping. She'll also be joined by John McLean, and he will be providing a little bit of a discussion after Stephanie has finished. So welcome her to the stage. Thank you.
Thank you, Michelle, and good afternoon, everyone. I'm here to talk about the giant willow aphid. I'd like to begin first by acknowledging the co-authors of this talk. So also present today is Steve Pawson. He's with Scion as well, and he's the research leader for our entomology group. He's based in Christchurch. Ian McIver is a research scientist with Plant and Food in Palmerston North, and he also manages the tree breeding program for the New Zealand Poplar and Willow Research Trust. Julia Martin is a summer student that worked with us this past year. She's with the Bay of Plenty Polytech. So first, just a brief introduction to this aphid. Like most aphid, it feeds on the sap of plants and secretes a great deal of honeydew. Unlike most other aphids, however, the giant willow aphid feeds on woody tissues. So its host plants are willows and poplars, and it feeds on the stem, not the leaves of these host trees. Looking at the image here, we can see a colony of giant willow aphid. All life stages are present. There are 5 life stages. What you can't see is a winged form. So just flipping back to the first slide, at certain times of year, you'll see more of these winged forms getting ready for dispersal. You can also see that the stem of this willow is covered in black sooty mold. So the honeydew secreted by the aphids is essentially like sugar water, and it fosters the growth of the sooty mold fungus. So these aphids are really good dispersers. They're thought to be native to Japan or thereabouts, but willows and poplars, their hosts, have been planted throughout the world. And we now find this aphid throughout the world. So it's on every continent now, except for Antarctica. And until very recently, Australasia was actually conspicuously absent. But it turned up in New Zealand, or was first recognized on Christmas Eve in 2013. In the coming weeks and that January, we began to receive samples in Scion's diagnostic lab. And together with MPI in that first season, we mapped the distribution of giant willow aphid in New Zealand and found it to be widely spread throughout the country. It also appeared for the first time in Australia and Tasmania in March of 2014. So oftentimes it wasn't the aphids that people noticed. They first noticed huge numbers of wasps buzzing around willow trees, and these wasps are feeding on this new resource, this honeydew that the aphids secrete. So here's some things you might see in the field, some indicators of the presence of the giant willow aphid. Looking at the yellow arrow, there's a German wasp feeding on the honeydew. Also bees are feeding on this honeydew. And you can see that the surface of the leaf the bee is on is covered in this black sooty mold. And more we're seeing entire willow trees and objects underneath the willow trees covered in this black substance. So this is the 3rd season now that we've had giant willow aphid, and more we're seeing these blackened trees. So I've brought to the conference a flyer that you can take away with you. There are some situated underneath the paper mache aphid over by the posters. Trees for Bees also has some at their desk, and we've got extras down here. This tells a bit of a story about the giant willow aphid, and also because the effects it's having in New Zealand are so complex, we've done a flowchart diagram on the backside to help you understand the magnitude of these effects. So I've just taken some snips from this diagram to put into the talk to help illustrate this. So beginning with the impacts that the aphid is having directly by feeding on its host plants. Essentially, the aphid pierces the stem of the willow, and the willow sap flows out through the aphid as honeydew. A little bit changed, but the host trees are losing a great deal of water and nutrients through these aphids, particularly because there are thousands, sometimes hundreds of thousands of aphids present on one willow tree. So this is causing decreased health of the hosts, And for the first time this year, we're getting reports in New Zealand of deaths of willows as well that have been severely infested with giant willow aphid. So we're seeing these impacts on willows, which is affecting the various uses of willows. Just trying to get the mouse to point to the circle here. So although some willows are considered weeds, by and large, they're very valuable to us here in New Zealand. So they're used to stabilize riverbanks and slopes, protect against erosion, and of course, they're a very valuable resource for bees, particularly in spring, providing pollen and nectar. So moving on to the effects that the honeydew is having itself. So the honeydew is present in great abundance. If you're standing underneath an infested willow tree, you can actually feel like a light rain falling down upon you. And so anything underneath becomes coated in this honeydew. For example, farmers are seeing sticky sheep, these sheep becoming dirty and creating extra processing costs for the wool. And then, of course, the sooty mold is growing on anything that's covered in this honeydew. For the plants themselves, so the willows and any plants growing underneath, they're becoming covered with this black substance, and they can no longer photosynthesize. Also, other things, anything beneath a willow will become sticky and turn black. So this could be, for example, your car or your patio in an urban environment as well. Now looking at other species that are really interested in the honeydew. So a lot of insects are out harvesting this honeydew. And we've mentioned wasps already. So wasps are a nuisance, of course, and wasps spell trouble for bees. And bees themselves are harvesting this honeydew. And this honeydew contains a sugar called melezatose. And when the honey is made from this honeydew, it creates a crystallized granular honey in the comb, and it's difficult to extract. It's called cement honey. And John McLean will be speaking after me, and he's going to talk about this in detail, so I'll leave that to him. I've also got I'll just move on to summary of these impacts. So it's affecting the host plants directly, which are valuable in our landscape and are important as resources for bees. There's a nuisance factor of the honeydew and the sooty mold. We're seeing increased numbers of wasps because of this new resource and the formation of cement honey. What I didn't mention in the diagram is that there are many more types of insects and even some honeydew-feeding birds, tuis and bellbirds, that are feeding on this brand new resource that's highly abundant. And so there will be some shifts occurring in New Zealand that have yet to be quantified in terms of biodiversity. Just to further illustrate the diversity of people being affected by this giant willow aphid, I've got a few cartoons. I'll just let you read them, starting with Cynthia, who lives in the city. Moving into the rural environment now. You could say Fred has bigger problems than Cynthia, but his problems are nothing compared to this real-life individual, our co-author. Which brings us to the solutions for giant willow aphid. So there are two solutions that have been identified that we feel are very promising. So one is looking for resistant willows, or willows that the aphid at least performs very poorly on, and biological control, so looking for natural enemies that will control populations of the aphid. Well, both of these are very promising. They're long-term solutions, so they'll require time for research and implementation. Also, nobody's doing this anywhere, so we'll be breaking ground to investigate these further in New Zealand. In the meantime, we'll have to look for some ways to manage the impacts. So just going through these, I won't say a lot about host resistance. This is something that Plant and Food are looking into. I don't expect you to be able to read this slide. I just wanted to point it out. It's a poster that's here now, presented by Trevor Jones, showing that they've already done some work to monitor the giant willow aphid in the National Willow Collection, which contains over 50 species and hybrids of willows. So they've been able to get some idea of signs of resistance occurring there. Trevor wants to hit the road today at about 3 o'clock, so if you want to catch up with him, please do so before then. biological control. This is something we're looking into at Scion. It comes in 3 forms. You can look for predators, pathogens, or parasitoids of your target organism. So looking at predators of aphids, ladybird beetles are well known. We've seen these feeding on giant willow aphid in New Zealand. And we've also seen this brown lacewing larva here. Overseas, there's a predatory midge that's known to be a predator of giant willow aphid. This beast in the upper right needs a special mention. This is the harlequin ladybird beetle, and it's brand new to New Zealand. It was first found only in April of this year in Auckland. Before you get really excited about it, because yes, it does like feeding on aphids, I need to say a few words of caution. It's an unwanted organism in New Zealand, so we may not spread it around the country. And it's unwanted for good reason. So it has been used historically as a biological control agent in several countries, but it has a habit of being a little overzealous, and it's outcompeted a lot of native species, for example, in North America, and it's become quite a problem in its own right. It's considered a pest species now. And one of the things it does is it gets so abundant in the autumn, and it looks for places to overwinter, and adults will come into homes in the thousands. So this is a nuisance, much like the brown marmorated stink bug is. And people are even developing sensitivities to this. So there's now something called human ladybird allergy because of this species. It also likes to gather in grapes and grape clusters just prior to harvest. So the grapes are harvested, the insects are crushed along with the grapes, it goes into the juice, and they taste really awful, and whole vintages of wine, particularly in southern Ontario, as an example, have been ruined because of this ladybird beetle. So I'm just going to leave that there and move on to a pathogen. So there's one known pathogen of giant willow aphid. It's naturally occurring in several countries. It does cause quite a dramatic drop in population late in the season. But one reason that we're not intending to follow through with this as an option is that it's not specific to giant willow aphid. I could say the same for the predators as well. They tend to be generalists, and a lot of non-target organisms would be affected. Fungi are also challenging to develop as biological control agents. They need to be isolated and cultured and put into some kind of medium that can be spread out into the environment. And once there, just the right conditions need to be present so that it can persist in the environment and do its work. So it's also costly and time-consuming to take this route. Just in case, though, something is happening here in New Zealand, something turns up, I've included this image which shows a related species on a different tree-feeding aphid. So what you would see is black bodies hanging from their mouth parts from the tree. So if anyone sees anything like this, please, my email is on this pamphlet. I'd like to know about it. So what we're most interested in pursuing is biological control with a parasitoid. There's one known parasitoid of giant willow aphid, and it appears to be highly specific, so that's exactly what we want. It's present in several countries already, and it's worth noting that in those countries, giant willow aphid doesn't seem to be a big problem, so it could be partly because of this insect exerting control on the populations there. So those are the long-term solutions, post-resistance on biological control. In the meantime, what can we do? These are just some suggestions. Giant willow aphid populations in New Zealand tend to peak in late summer and into the autumn. So knowing this, I've already heard that some people, in fact, are taking the honey off their hives sooner. Another option would be perhaps moving hives away from willows prior to this peak abundance of giant willow aphid. And if wasps are your problem, wasps can be controlled directly. So there's been some preliminary work done already. This is a slide from Ian MacGyver of Plant and Food. This work is not to control the aphid. It was some preliminary work to get some hard numbers on the impacts that the aphid is having on willow trees. So it's a spray trial. It was conducted at 2 sites. If you look in the image, you'll see some blue dots and some yellow dots. So these represent trees, and the blue ones were protected with insecticide. In the graphs you can see P for protected, and the blue bars. The yellow ones were not protected. So trees were measured for height and collar diameter, both before and after insecticide sprays. And they did find significant differences at both sites, where the trees that were protected against the aphid did grow more. With collar diameter, the results were not significant, but you see the same trend. They did better when they were protected with the insecticide. At Knox Estate, something unusual happened. These woody plants seemed to lose mass. It sounds a bit unusual, but the same thing has been found in a study on willows in the UK that were infested with giant willow aphid. They actually appeared to lose their woody tissue. So some work that we've done at Scion. Thanks to the Bay of Plenty Regional Council, they let us do some work at a willow nursery in Whakatane. So we had 3 objectives. We wanted to monitor the population of the aphid over the season and link this to the presence of melezatose or cement honey, and also look for interaction with other insects at the site. So we chose 2 willow hybrids, 8 trees of each, and we marked a branch on either side of the tree, divided that into 3 segments, low, mid, and upper crown. And we, means Julia, our student, came weekly and counted the individuals in those sections of branches. We also tried to estimate the % coverage of stem by the colonies of aphids, and we found this really difficult for a few different reasons and ended up dropping this. And we placed water-sensitive cards out under the trees periodically to catch the honeydew falling down. So these cards are yellow in color, and where the honeydew droplets fall, it changes the color of the card. So here's a row of our study trees with the cards placed underneath. And you can see the splatter that's formed on the cards by the honeydew. You can use software to calculate then the area covered by honeydew on these cards. Unfortunately, these results didn't correlate well with our count data. We just felt that we didn't put enough out. It's actually, I think, quite a good system, but you need to do it more frequently and with more cards. What we did find, we were able to extrapolate, knowing the area covered of the cards that we had, what kind of square meter coverage of honeydew was occurring in a 2-hour period. And under these small, they're only about 2-meter willow trees, we had up to 5 meters coverage of honeydew in just a 2-hour period. So big stress on these trees. In terms of growth of the colonies of giant willow aphid, this graph is broken into 3 sections showing the base, mid, and tip of the trees. Looking at the clone, the hybrid shown in green, the peak abundance in all sections of stem, particularly the mid-stem, occurred in about March. And by early June, the population had really dropped off on this particular hybrid. And this was in stark contrast to the other trees at the site. This other hybrid showed an increase in abundance over time and the insects moving up higher and higher into the tip of the crown. It was worth noting that at this site, the tree with the most aphids at the end of the season was the one that still had almost all of its leaves. So there was a physiological difference between the 2 trees. This image just shows aphids clustering at the base of one of our study trees early in the season. So this is about a 6 or 7 diameter stem. And also fairly early in the season, some aphids beginning to move up more into the tip of the trees. You can tell these haven't been there for very long because there's no honeydew present yet. So in terms of linking the population of giant willow aphid with melezatose in honey, we haven't yet analyzed our honey data. So this is to be continued. I'd like to acknowledge Jason Stanley in the image. He's standing to the right of John McLean. He brought hives onto our site and helped us with our honey sampling. He also took this picture in which you can see we were getting a bit of melezatose. There's some crystals forming in these uncapped cells. We didn't get a lot obvious at this site, and we think that's probably because Whakatane town was just right across the river, so bees were probably foraging there as well. And which brings me to the other acknowledgement. Craig Sinkinson of Coastlands Apiaries normally keeps his bees at this site and was kind enough to remove them for this season so we could do this study. In terms of predators, we found all 3 of these ladybird beetles feeding on the aphids, and we found several types of honeydew feeders. So lots of wasps and bees, a couple of ants, some flies, and even these 3 ladybird beetles appeared to be interested in the honeydew as well. So all of this preliminary work that we were doing and that Planet Food were doing was done while we were eagerly awaiting the results of an SFF bid. So this bid was led by Apiculture New Zealand, with Scion and Planet Food as the research providers, and with co-funding pledged by Apiculture New Zealand, by the Poplar and Willow Research Trust, and by regional councils. We also had in-kind support from several organizations and individuals. And we applied in August last year, and this April, we got the bad news that we weren't successful. We are going to be reapplying, and we're feeling really good. Apparently we came quite close, so we're feeling very positive about applying a second time. But unfortunately what it means is it puts us a year behind. So if we had been successful, we would be receiving funding this July to carry on. Now we'll be looking at next July. So in the meantime, just speaking from Scion's perspective, we intend to forge ahead. So we're going to be applying to the Environmental Protection Authority to import this parasitoid. We're working on collaboration with overseas partners where the parasitoid is present so that we can go and collect it. And we're also going to be developing laboratory colonies, not only of the giant willow aphid, but of the other aphids, native aphids, that we'll need to test this parasitoid against in New Zealand. As part of the bid to the Environmental Protection Authority, as part of that application, we'll have to do an economic impact assessment, and we're not sure about some more field studies. So Scion is contributing core funding in order to do this, but it won't be possible unless we have co-funding as well. So we're hoping that we'll have some extra support, and we'll be able to get these objectives done, and that come next July, we'll be this much farther ahead and able to hit the ground running. That brings me to the end. I'd like to thank Apiculture New Zealand for the opportunity to speak today. Thank you, and I'm handing it over to John McLean.